Hello, my name is David Allis. I'm a professor and head of laboratory at the Rockefeller University in New York. My laboratory studies chromatin biology and epigenetics. If you were able to watch my first iBiology talk in this series, I was trying to introduce what is epigenetics and give some of the historical perspective, some of the big experiments that led to sort of the concept that there may be something more to our genome that led to our concept of epigenetics. Today, I hope I can build on sort of some of those thoughts and tell you about how does epigenetics impact some of development and disease, focusing a little more on some of the more current experiments that are ongoing in my laboratory and others. So as again, the same sort of orientation would be epigenetics as a term, something in addition to our DNA, on top of DNA, traces back to Conrad Waddington, who in the 1940s coined the term to try to explain how in development would a young embryo sort of ultimately pass through development and give rise to many different cell types, whether it's blood, nerve, skin, whatsoever. And once those sort of cell identities have been established, all from the same genome, then the question would be, how could that happen with the same genome? So he was fascinated by that process. Of course, once the differentiated cells have been established, in general, they know who they are and what they're to be. So blood cells will give rise to other blood cells and so on. But every once in a while, so there's a memory aspect to it, but every once in a while, unfortunately, some of these epigenetic landscapes get confused and give rise to sort of cells going backwards in development where they become more undifferentiated, more proliferative, and the outcome of that often is a cancer of some type. And I'll talk a little bit more in my lecture of some of what we're doing that has a sort of a cancer connotation. So to give you some insights, I have to explain how the genome is packaged. If you look at this cartoon of a eukaryotic nucleus, you can see that although we're depicting some naked DNA, in fact, very little of our genome is naked Watson and Crick DNA. There's a fundamental repeating spool, if you will, that you can see in the lower right-hand corner is now at atomic resolution, thanks to Tim Richmond and Carolyn Luger. That fundamental repeating unit, like a bead on a string, is called the nucleosome particle. And if you follow up on the cartoon, we don't understand quite in the field all the details of how these nucleosomes pack and pack and pack to give rise to higher order structures. But ultimately, 2 meters worth of DNA have to package into a eukaryotic nucleus. And I think it's intuitively obvious that there's going to be regions of the genome that are reasonably inaccessible, and there's going to be regions of the genome that are accessible. So let's take a look at the nucleosome in even higher order magnification. So I think this is a very elegant structure. This was a landmark structure in 1997. And I just use this cartoon to sort of ask you to notice that emanating outside of the DNA are histone, what we refer to in the red box as tail domains. And you might be tempted to think, well, they can't be important. They're not really packaging the DNA. And in fact, you can set up situations in the lab where you can chop the tails off with either enzymes or with genetics. And indeed, you still make a very good nucleosome particle. So why has nature held on to the tails being as conserved as the rest of the histones, where in fact, if we pick on the blue histone, which is sort of going to be the star of my show, histone 3, the tail of histone 3 is conserved essentially from yeast to man. So if it doesn't package the DNA, why are these tails being held so tightly in evolution? Well, the answer will be that those tails are a very attractive landing pattern for covalent modifications, something that I talked a little bit more in my first iBiology lecture. But if you pretend that there's a covalent modification by the red ball, this could be an acetyl group, this could be a methyl group, it could be other modifications. We now know that those modifications are either added by writers or subtracted, taken off by erasers, or even in some cases, there are post-translational modifications where protein, we'll call them readers, can bind to the modification and engage the histone chromatin fiber that way. And so I think we now have lots of insights from many laboratories of molecules that fall into these categories, in the writer category, the eraser category, or the reader category. And many of these have become attractive drug targets. And proven to be quite useful in clinical settings, in part because the DNA is not altered. So this may not be fair to call this a reader, but there would be a very powerful cell biological tool that I think my laboratory and others have put to good use. And that would be, can you find an antibody molecule? Can you generate an antibody molecule that might be highly specific for the modification of the post-translational modification on the tail domain? And I want to give you some spectacular views of where these type of antibodies started to let the field know that there's a lot going on here with this epigenetic regulation. This is a classic cover of a Cell article that was published in 1993 by Brian Turner and his colleagues, where they generated an antibody, I'll refer to it as anti-acetylated, so that would be the chemical modification, anti-acetylated H4. And they spread mammalian chromosomes out on a slide. I think you can see on the left there's a lot of kind of green staining chromosomes. And they made note with a counterstain that was enabling them to see sort of the underlying non-antibody stain, that one chromosome in the dashed box with the circle, arrow pointing to it, that there was one chromosome that was lacking the green stain, the yellow-green stain. And that, in fact, turned out to be the X chromosome. And I have to tell you, for completeness, these were female mammalian cells. So they were now exhibiting a phenomenon called X inactivation. This is one way the two X chromosomes in a female will be kept sort of equivalent genetically to the single X chromosome in the male. And here's a higher mag, just so you can see how really spectacular this was. This is the same image just blown up. So you can see it's really quite clear that that inactive X chromosome labeled XI is really not staining with this antibody. This is an assay we refer to as immunofluorescence. And I think if you look carefully, some of you may notice that some of even the chromosomes that are largely yellow staining have little zones where you see the underlying red. And in fact, those are regions where we don't have much acetylation. And of course, the correlation here is that when you don't add these acetyl groups, in general, these genes, and in this case a whole chromosome, are not transcriptionally active. So hypoacetylation tracks gene inactivity. Now you might say, well, maybe every methyl or acetyl mark is created equal. Well, here's the real sort of amino acid sequence across the top in blue of the histone 3 tail. It's not the entire tail. This is in one-letter amino acid code. But I'll just ask you to look at 2 lysines. I should say that's where these chemical groups are often added, a lysine residue, that's K. And so you can see if you've generated an antibody to K9, you would be able to sort of potentially get an antibody that might read the red sphere, if you will, a methyl mark. And I'll just tell you that that's embedded in a sequence, too much detail, ARKS. Now you can see if you extend down and look further into the histone 3 tail, skipping some residues, you'll see another ARKS motif in the yellow box. Again, that's a lysine at 27 now, so we're further into the tail. It was known to be methylated. We could generate an antibody, we and others, to the lysine 27 methyl. And I need to sort of remind you of a silencing paradigm that came up a little bit in my first iBiology lecture. When you want to silence genes and you use methylation, you ultimately have to remove pre-existing acetyl marks with histone deacetylases or HDACs. That sets the lysine up where it can now obtain a methyl mark by histone methyltransferase, a writer. And then there are reader proteins, X and Y, that are being dictated here that read these methyl marks. So the question is a K9 methyl mark identical to a K27 mark? And here's some real, again, immunofluorescence staining patterns. This takes advantage of the Drosophila has a beautiful set of chromosomes. They're called giant polytene chromosomes. All you need to know is that they're big and they're huge. And you can spread these chromosomes out on a microscope slide. And on the top 2 panels shown in red, you can see the staining with either a K9 methyl antibody on the left or a K27 methyl antibody on the right. And I hope you might agree that they don't look at all the same. And in fact, what we know is the sort of genetic epigenome area on the left where there's like a central blob of red staining. Those genes, that genome sequences are really heterochromatic and always off. In contrast, the upper right red panel with the K27 methyl staining is genes that are known to be out on the chromosome's arms, that should be clear, and they're regulated off. They're not always off, they're regulated off. And below each panel are antibodies that were developed to the corresponding readers. It's too much detail, so on the lower left, we'll just consider it X. On the lower right, we'll just consider it Y. But I think you can see there's a pretty good agreement of the reader sort of tracks and co-localizes with its mark in either of the two K9 versus K27 situations. So this is sort of like a way to look at the genome in Drosophila. Now, yeast is an organism I want to set up carefully later in my talk, but I want to say, but are all methyl marks silencing in nature? No, they're not, and in fact, if you look at the very tip of the H3 tail, lysine 4, that was shown by many laboratories to be an on mark. So not all methyl marks are silencing, some are activating. But yeast as an organism has this amazing property that you can obtain genetic knockout strains. Not all of them will kill the organism, so you can get viable yeast strains that have knocked out individual genes. And all you need to know for this experiment is the methyltransferases, the writers that add methyl marks, they all have a domain called a set domain. That's too much detail. But then once the yeast genome was sequenced, we could ask, well, how many set domains are there in yeast? And the answer was 7. And so you could just order knockout strains for each of the individual set domains, set 1 knockout, set 2, set 3, set 4, and so on. And then just again, take your favorite antibody, in this case a K4 methyl antibody, and just do what we call a Western blot. The lower panel is like a loading control. You can sort of see, did we load fairly? And you can see sort of a nice even staining. But I hope you'll agree with me in the green box with the downward pointing arrow. There is no strong staining for the lysine 4 methyl mark in a set 1 knockout strain. And what's relevant about that isn't then just that told us that that's the writer that adds the K4 methyl mark, but by computational inspection, we were able to determine that set 1 is equivalent to a fly gene known as trithorax, TRX, and more importantly, that's equivalent to a human gene that's known as mixed lineage leukemia, MLL, and that's a cancer leukemia causing gene that is, its misregulation is in part tied to a sort of establishment of K4 methyl marks. So, so you might have said, well, I don't really care about yeast, I don't really care about flies, why is Allis telling me all about this? This was the type of experiment that suddenly got the field interested in K4 methyl marks as having potentially a disease implication, in this case, mixed lineage leukemia. Another important thing I want to sort of end on with my antibody part of my talk was antibodies enable you to inspect the genome not with pretty pictures, not with Western blots. You can take the genome, shown across the top panel, and fragment it. You could fragment it, shear it with various different methods, and basically break the chromatin particles, those nucleosome units, into little bits and pieces after a cross-linking step. Well, of course, histones are right up with the DNA. The green sphere might not be a histone, it might be a transcription factor. It might cross-link as well. And then your sonicated, fragmented material gets incubated with your favorite antibody. So in the third to the bottom panel, you might be able to take an antibody against one of these modifications or an antibody against the transcription factor and do a precipitation. And then once you've recovered that material, you can get rid of the antibody, the protein, and sequence the DNA and find out where in the genome was this particular transcription factor or this particular modification. If it was on a histone, where is it? And I'll go back to the sort of now electron micrograph spread chromatin, and you can see again the top panel, the nucleosome, we'll call it thin filament, the bead on a string. It's a very famous pattern. and then much more on the lower panel, condensed chromatin. And I'll just say through genome-wide chromatin immunoprecipitation experiments like what I just showed you, they're often named CHIP experiments. That stands for chromatin immunoprecipitation. Many researchers have now asked where in the genome are different marks. I don't really think it's important that you look carefully in the green or the red boxes, but I'll just make a quick comment. There are marks that correlate with enhancer elements. There are marks that correlate with promoters of genes. There are marks that correlate with active gene bodies. So on the active side of the coin, these modifications do set up a pattern. A long time ago, Brian Strahl and I referred to this as a potential histone code that's telling different chromatin DNA elements what their property might be. In the red box, as I sort of illustrated with my Drosophila chromosomes, there are permanently silenced genes in heterochromatin, and then there are repressed genes that are repressed on demand, again, all with different marks. So it's very exciting that now researchers have gone through everybody's genome, from normal cells or from abnormal cells, cancer, non-cancer cells, and seen that much of the epigenetic landscapes are altered, suggesting that this may be part of the disease mechanism. Now, at this point, I may have led you to believe incorrectly that a histone is just a histone is just a histone, and that there's modifications that alter the histones, and there could be writers and erasers and readers. But what about the histones themselves? And it turns out that nature has been very clever in creating a major histone. Let's take the histone 3 family as an example. There's the major prototypical histone 3. It packages the chromatin nucleosome in a major way, pretty much goes down the whole chromosome, and you know, that's the textbook histone 3 that helps to do that job. But nature has created some histone variants. We refer to them as variants. They weren't very well studied, and in the mammalian system, there was a histone variant 3.3. Now that might be annoying to some of you. There's a 3.2 and there's a 3.1. But for the simplicity of this talk, I'm grouping the 3.1 and the 3.2 together as just stage 3. So a question for those of you who are listening is, well, how many amino acids different is 3.3 from the regular H3? And the answer is, depending on who you compare against, the answer is only 4 amino acids or 5. So it's almost a regular H3. But others have found, Steve Henikoff in particular found, that if you follow pathway number 2, 3.3, the histone variant, was being selectively targeted for active genes. It didn't go down the whole chromosome. It actually used its own chaperone, its own delivery system, if you will, that's labeled HERA, to be targeted to active genes. Well, that was quite remarkable. You know, you had the regular H3 going elsewhere, and 3.3, this little minor, a few amino acid different variant going to active genes selectively. But then Aaron Goldberg joined my lab, and he was interested in 3.3 as well. And he found remarkably that 3.3 had a whole nother personality, and that's shown in the pathway 3. There was a different chaperone named DAX, and believe it or not, it was taking 3.3 to the ends of chromosomes, or the chromatin right around the centromere. That's that constriction on the chromosome below. And so how could the same histone variant be going into active or inactive? And part of the answer seemed to be with different chaperones. Now, I didn't show too many of my lab members, but this is relevant. Aaron Goldberg was the student. He's in the lower right-hand corner. He was in the MD PhD program at Rockefeller, very talented student, and he was the one who developed the DAX connection to 3.3, the targeting. And I guess when you get a paper published in Cell, they invite you to make a cover. And what always blew me away was the woman on the top, Yifan Xu, was also one of Aaron's fellow graduate students in the MD PhD program, close friends. And much to my surprise, talk about talent with your students, she painted that Cell cover. It didn't get accepted in the journal, so that's the bad news. So it never saw the light of day in a publication. But I was always very impressed there were these kinds of, in this case, both MD, PhD students, and she had this hobby of being an artist and a painter. And I've always thought that her artist drawing of the Dax individual, the gentleman in the left who was putting nucleosomes into silent chromatin with Dax, happened to be maybe not just an accident that he was in a white coat. And you might say, well, where's the disease? Because Aaron was going to go on to become a physician scientist. Aaron's paper was published in 2010. So if you say, well, why do science? Well, totally without our knowledge, the next year, 2011, this paper was published in a high-profile journal, Science. We knew that this was a big cancer group, largely at Johns Hopkins in Baltimore, and they were sequencing human tumors for a deadly cancer known as pancreatic neuroendocrine tumors, or PANNET, and they found a remarkably high frequency of genetic mutations that were in DAX and this other partner of DAX I haven't spoken about, ATRX. And the frequency of these mutations was quite high. These authors really weren't chromatin biologists like my lab, and they ended up writing their paper saying, I wonder what this has to do about chromatin. Of course, we had been studying DAX from the chromatin side of the coin through Aaron's work. And to put a celebrity face on the disease, this type of tumor is what Steve Jobs, the founder and creator of Apple, passed away from, sadly, in 2011. So, of course, as a biochemistry lab, we were interested in finding out, well, if there's only a small number of amino acid differences between 3.3, look at the top sequence on the bottom cartoon, and the regular, if you will, H3. I'm only showing you the amino acids that differ. And remember, I told you there's only 4 or 5. We were able, and the we here is Simon L. Sesser and colleagues, were able to crystallize the structure of human DAX, and found out that it actually really is the specificity factor. DAX binds to and likes 3.3. Now what I'm showing you in the right are a couple gels. Now admittedly this is something I won't describe super well, but they're called pull-down assays. So we actually asked DAX to sort of incubate with histones and then pull it down with an antibody and then run it out on a gel. And I hope you can see that there's 2 prominent bands that are at the position of roughly H3 or H4. I should say that H3 always partnerizes with H4. Well, let me ask you to look at the left-hand side first. This is when you take DAX, the protein specificity factor, and you incubate it on the left-hand most panel with wild type 3.3, the regular 3.3. I hope you see a prominent band of H3.3 and H4. But now, for fun, Simon asked, what if I take that last glycine? Now that happens to be where the red arrow is, pointing downward. It's a glycine, that's an amino acid that's very small. And I'll make that glycine become the methionine, that's the M. I'll give it an M it never had. And I hope you will agree in the little dash box, there's no more binding of 3.3. And then Simon did the mutagenesis game in the other direction. He said, well, on the right-hand pair of 2 gels, let me now start with the regular H3, and the leftmost lane there, you'll see no binding. It doesn't bind. It's remarkable to me. DAX doesn't care about regular H3 until, on the very rightmost lane, you give the regular H3 that glycine it never had. So in that last red box, now if you take methionine at position 90 and make it a glycine, I hope you'll agree in the red box there's 2 strong bands. So we would have never predicted that something as trivial as a glycine mutation to methionine, 1 amino acid change, would be part of the specificity recognition, which leads me to something that people make fun of me for saying, does every amino acid in histones matter? And I should have mentioned, but on the original disease paper, there was a middle author. I won't go back to the slide, but Laura Tang was an embedded middle author on that paper. She's across the street at Memorial Sloan Kettering. Laura, we've teamed up with to look for then where are mutations and what are the properties of these mutations in real patients that have pancreatic neuroendocrine tumors, or PANNETs. On the left-hand side of this display are gene expression profiles. They're just different genes from top to bottom. You won't be able to read them. But in general, on the very bluemost genes, these are expressed or non-expressed genes that are coming from tumors that came from the patients that have been sequenced and genotyped to not have a mutation in DAX. They had a tumor, they had trouble, but they didn't have a DAX mutation. And since red in this highlighting is expression, I'll just tell you in the upper left-hand quadrant, the proteins that are most strongly expressed are pancreatic proteins. On the right hand, from the vertical bar to the right, are tumors that were identified, pancreatic neuroendocrine tumors that came from LAURA, both primary and metastatic, where they were sequenced to have the mutation in the DAX protein that I've been talking so much about. And I hope you agree it looks quite different, and now the vast majority of the highly expressed or red genes happen to be liver proteins. So I think what's happened is the cell has become confused. The cell was meant to be a pancreatic cell. It switched its, if you will, epigenetically driven identity, and now it's taking on liver properties. And that may not be a coincidence with the fact that when this tumor originates in the pancreas, its primary site of metastatic lesion and ultimate death for the patients is it travels to the liver. So this is a case where something as seemingly as unimportant, if you will, as a histone variant, a few amino acids difference, has now been directly implicated in a human disease. And this is ongoing unpublished work in my laboratory and others with Laura Tang. Now, I've mentioned that, I've made fun of myself here saying, does every amino acid matter? Maybe from this DAX example, you might be convinced that there really is something to that. But how would you really test that as a scientist? So this will seem like an aside to you, but I'm going back far away from human biology, back to the yeast model system. This is budding yeast. This I want to pay credit to Michael Grunstein and Mitch Smith and others. And in the cartoon of the budding yeast cell, I want you to notice that there's a very small number of genes that are meant to be chromosomal copies of the 4 core histones. I don't know if I mentioned there are 4. And yeast is remarkable in that it only has 2 copies each. So there's 2 copies of each of the 4 core histones. And Grunstein and Smith and others realized that they could delete completely all of the chromosomal copies of those histone genes. Now, of course, a yeast cell wouldn't be alive without histone proteins, and so deleting its genes would be incompatible with life. You have to package your genome. You have to package it with histones. But that cell could be kept alive by the circular molecule, which we refer to as an extrachromosomal plasmid. And it's keeping the cell alive because it has 4 copies, the blue, the green, the red, and the orange, of a wild-type copy of each one of the 4 core histones. So that plasmid will keep the cell alive. And then the brilliance of this genetic system set up by these researchers was they realized that you could do this trick of what we call a plasmid shuffle, where you could then mutagenize a plasmid on the left, the one that's marked with a lysine 2 marker, and that could carry a mutation of interest. So in the yellow box, I'm giving you an example that the blue histone, I've been focusing a lot of my attention on histone 3. What if we took its gene and we mutated one of the special lysines, and you mutated it to an arginine? Some of those of you who know your amino acid will know that a lysine for an arginine keeps the positive charge, but a lysine is one that can only be acetylated, for example, not an arginine. So a K to R mutation would be a great test of whether that lysine matters. And then you can shuffle that plasmid in to the cell, and under the right sort of selection pressures, you'll lose the original wild-type plasmid that was marked with URA3. So this was an elegant test. I used to tell students, if you wanted to do histone genetics, you better do it in yeast, because this was one of the ones that you could do real genetics on. So the big surprise for my laboratory came in the close of 2012, when 2 groups, almost back to back, Nada Jabato at McGill University and Susie Baker at St. Jude's, studying pediatric tumors, and the most devastating type of pediatric tumor, a brain tumor that's essentially universally fatal, found that the mutations in the patients mapped to histone genes. Not just one copy of the histone, not just all the copies, just one copy of the histone gene. You inherit histone genes from mom and dad. Those are called alleles. And you have many, many histone copies in the human. So what was remarkable is just one histone, one allele, was carrying a mutation. And even more remarkable, let me show you where the brain tumors arise in these children. For the experts, it's been known as DIPG, it's easy to say. That stands for diffuse intrinsic pons glioma. The tumors occur in the base of the brain. It's called the pons, hence the DIP. And essentially, it's an inoperable, untreatable disease. These children are usually diagnosed around the ages of roughly 5 to 8, and they pretty much don't ever make it, and sadly die within about a year and a half. And so these DIPG tumors had no molecular understanding, no therapeutic potential, and here the mutations were mapping to histone proteins. Now it turns out not just any histone protein, they were mapping to this histone 3, which is why I'm making it the star of my show, and then they weren't mapping to just anywhere in the histone, they were mapping to the residues, the lysine residues. So as much as I was sort of offering to you yeast genetics as a powerful tool, this is a case that I would have never bet my money on, that now the genetics is coming from the human and closely associated with a deadly disease. And again, in the histone tails, these mutations were mapping right at the point of attachment, right where that modification lollipop comes off. The base of that lollipop is where the mutation maps. And too much detail perhaps, the lysine is always mutated to a methionine. So I might slip up and say K to M. So of course we'd like to know what is this, as a chromatin lab we want to know what these modifications are doing. It gets more human for me because I've had the tragic pleasure, I think, to be able to give seminars where DIPG researchers like Michelle Manji at Stanford introduced me to families under her care, in this case the Krantz family in San Francisco. And I wouldn't show you this except for this family made a public documentary of the tragic death of their daughter Jennifer, who was diagnosed with DIPG. Michelle said to me, Dave, we got to get on top of this. I don't want to do any more of these tissue harvests. And sadly, after I visited Stanford, Jennifer did pass away, and aspects of her brain tumor were sent to my laboratory in New York. I was born and raised in Cincinnati, Ohio. That's irrelevant to you. But I was stunned, really, when in 2015, the national news covered this very brave woman, Lauryn Hill, born and raised in Cincinnati, Ohio, and she was diagnosed, this is atypical to be a 19-year-old, but she was diagnosed with DIPG. What she wanted to do more than anything else before she passed was to shoot a college basketball hoop for her team, and she actually did that and raised an incredible amount of money, over $1 million for a cause called The Cure Starts Now. So very inspiring sort of national sports story where a woman died from DIPG, albeit older age than normal. So of course my lab would be interested then is we know what some of these lysines are doing. Certainly if you listen to my first biology lecture, you'll know that some of them are sites of acetylation, methylation, who are the writers, who are the, and I just want to tell you that as the cartoon sort of shows, we were stunned to realize that what might be the problem with a K to M mutation was that we might be impacting the methyltransferase writer. And I'll give you one experimental example on the next slide. So these are Western blots. I think you can judge some of the bottom of this gel are just the histones stained and various other loading controls. But let me ask you to look at the top row. The top row is probing the blot with a methylated antibody against lysine 27. That was the position where the founding DIPG mutations were identified. And I think you can see that if you're able to read the K27M box, that's the rightmost box, there's no staining there. Essentially no staining there. It's like a little hole. And Peter Lewis, who was a postdoc in my lab who did this remarkable blot, he then asked, is it magical that it's a methionine? So to answer that question, he altered the lysine to every possible amino acid. So I've loved to nickname the alphabet experiment. And I think you can see, based on the top row, none of the other amino acid substitutions cause a problem, except for the second leftmost box, which is a lysine 27 to isoleucine, or I. So at this point in time, we only knew that there were 2 amino acid changes that cause a problem. K to M, that was the founding mutation, and now, although there was no cancer for it, K to I at position 27. As well, sadly, we were able to stain, back to this antibody staining immunofluorescence, we were able to stain brain samples from tumors like Jennifer's. And on the top of this stain is an autopsied brain sample from a patient that succumbed to a brain tumor that was not from a patient that had DIPG and did not have the K27M. And you see there's a lot of staining, a lot of brown cells that are staining with this methyl 27 antibody. But on the lower panel are samples of staining from autopsied children's brains that came from, like Jennifer Krantz, a DIPG patient who did have the K27 sort of a mutation. It's just unbelievable to us. There's essentially no staining of this methyl mark. And if I go back, whether it's the alphabet and you're doing a blot and you see a little hole on the blot, or maybe more relevant, staining real human tumor samples, there seems to be a dramatic drop in the signal, suggesting that even though these patients have tons of wild-type histones, there's something going wrong. And we now know the problem seems to be in part tied to the writer. This mutation is inhibiting the writer. So quite remarkably then, if you will, the mutation in this tail, and I want to be clear, this is a genetic mutation. Some people say, Allis always likes epigenetics. No, no, no. This is a real genetic mutation that now is impacting the epigenetic machinery that now is somehow, if you will, poisoning, hence the skull and crossbones, the writer that puts on this mark in a dramatic way. And of course, that may confuse the landscape enough to cause trouble that ultimately leads to the misregulation of genes and a patient in a lethal situation. I would have never predicted this, but now if you follow the little asterisks, if you will, other lysines in the tail, so a green lysine, if you will, mutated, has now been found to correlate with pediatric blood cancer. If you go further in the tail to the brown color, there's a deeper lysine in the tail that's now been found to be mutated to methionine to bone cancers. So it looks like the H3 tail is somehow peppered with different mutations, almost always K to M, that are causing different pediatric cancers with remarkable cell specificity. And as a footnote, I'll just say that if you transplant these cells back into the side of a mouse, if you transplant a wild type cell, like the left animal, you don't have any tumors formed. And if you do the same experiment with the same type of treatment to a mouse on the right, in this case it was a particular lysine mutation, K36M, you see there's really very pronounced tumor formation. This was a tumor that tracked the bone, and this transplantation is not even taking it to a bone, it's just the side of a mouth. And so it's really a very striking demonstration that these we've nicknamed these mutations now oncohistone mutations, sort of as a buzzword, but this suggests that they really are driving cancer in this situation. There is no other genetic mutation other than these histone mutations, and the perplexing part is they have lots of wild-type histones, so why is this even a problem? And so that's fascinating to us. So let me sort of wrap up here and give you a couple sort of now emerging concepts from my lab. I've made several references to does every amino acid matter? Well, I think unquestionably now you might have taken the yeast example or some of my other examples, but I think these oncohistone mutations, so this is too much detail, but this is the portion of the H3 tail where some of these K mutations have been found. And I just want to set the record straight. At the lysine 36 part, that's the green lysine, not only have K to M mutations been found in the patients, this is the bone cancer, but also K to I. And that was something we saw in our blots. So it's not just pie in the sky anymore. It's not just K to M, it's also K to I. So as I said in the bottom of the slide, our current and driving hypothesis, what will make me as fulfilled as I could possibly be as a researcher, is can we learn our knowledge of histone biology and all of this epigenetic pathway, can we bring this to bear to learn how to detoxify these mutations to try to get around some of these devastating pediatric cancers? It's sort of meant to be a laugh and some comic relief, but you come to New York, I work in New York, some people like the Yankees, I'm not a baseball person, but in fact, I want to leave you with the impression, don't rule out the Mets. And of course, by Mets, I mean not the baseball team, the Mets, I mean these methionines, because that's the amino acid that seems to be causing so much trouble. And these K to M mutations then are really driving quite a bit of the research in my lab, because I think we view them as a tool that we can now use to sort of perturb each of these lysine-driven pathways, selectively, if you will, poison them at will, and then ask, what is the downstream biology that's affected? I would be remiss if I said that it's all just cancer, cancer, cancer. This isn't meant to be something that you'd really absorb, but it's a nice compilation of papers from the recent literature that have now said, not so much how does epigenetics impact cancer, what about single gene diseases, what about neurology, what about fibrosis, what about cardiology, immunology. And if you were able to read the titles or read these abstracts, you'll find that in all of these different non-cancer diseases, it seems as though the problem is in an epigenetic pathway that's being misregulated. So Jim Watson said this quote that I think is pretty remarkable at this point in time. After listening to all this epigenetics play out, and of course he was one of the Watson and Crick founders and discoverers of the double helix, you can inherit something that appears beyond the DNA sequence, and that's where the real excitement is now. This is my cartoon wrap-up. If you watched the first iBiology talk of mine, you saw this before. I think it's a nice summary, though, that there's real genetics on the left. I think to pick on my oncohistone, what I just talked about, those are real mutations. Hence the red asterisk. Those are real mutations in the DNA, but unquestionably, and in this case, even in a gene that encodes a histone, but they're impacting on the right side of the coin, the slide, the epigenetic machinery, and in this case, this specific case, the writer of an epigenetic methyl mark, and these sort of interplay of different modifications of histones, different modifications of DNA, they are reversible, and that's the hope. We can maybe then reprogram and get around these mistakes that are causing some of these disorders. So if you're really fascinated about epigenetics and want to read more on the topic, this just happens to be a new textbook on epigenetics that was put out this past year by Cold Spring Harbor Press. And it helps me to sort of, I participated in this exercise with some wonderful people that are the editorial team. And I think from Thomas Genuine on the left, Monica Lochner, Danny Reinberg, this was in Danny's office in New York, myself and Marie-Laure Caparas, all of us felt passionate about trying to bring this together in a textbook form so that this complication and all of this complexity could be slowly digested by those folks who want to read more about your genome from the standpoint of the epigenome. Thank you very much. And I have a contributor slide. I'd like to certainly pay credit to where if I talked about anything that was sort of experimental or laboratory related, I showed a few pictures, but I have to sort of pay credit to all the wonderful students, postdocs, and techs that have been in my lab over the years. You might have gotten a sense that we like to collaborate. We do. For example, the physician scientists with whom have given us tissue samples from patients. iBiology, the project, shout out to Ron Vale for getting me involved with this, and others. The Lasker Foundation for hosting me today and the filming, the crew here, my funding, and Rockefeller University, and thank you for your time and attention. Thank you very much.